So the background to this conversation is that I periodically log into Apple's podcast utility, and I logged in recently, and it indicated a number, which I probably should have already known, that people were still listening to these Biota podcasts, Biota Live, Biota conference audio, a bunch of additional stuff that I have just available. And I asked a bunch of listeners who they'd like to have back on, and somewhere in that list was Bruce Damer, so I'd like to welcome on Dr. Bruce Damer.
Thank you, Tom. It's a pleasure to be back after at least 4 years or so.
It's been a while. I seem to recall we had a conversation. I'm not sure whether we'd left San Jose. I think I was still standing in my house in San Jose, so it's probably got to be at least 2 years ago. And you at the time were just wrapping up, I think, was it called Project Moonshot? And returning to a new Biota Institute, which I saw recently from video. You're now working with SETI, which seems to be a very good linking. But for folks listening in, can you recap what the past 4 years have been like?
Yeah, perhaps the primary thing was I met my now wife and life partner, Catherine Lucas, who is now Catherine Lucas Damer.
Congratulations.
And we got married last August 29th and went out to Burning Man and got blown out.
Very good.
But that was really significant, and we made home together. I had just finished building my Gandalf house here, and it was just filled with sawdust, and she helped. And we worked together and made it into a home, a true home, and we now have our own beautiful shared life and shared projects.
Very good. So in terms of the Biota Institute, the return to the Biota Institute in some regards, I mean, it has a different vision now, but it very much was based on the EvoGrid work. Can you kind of walk through what's gone on with that?
Yeah, so as the listeners, the very long-time listeners would know, I was finishing my PhD in 2011 with this evolution grid or EvoGrid project to simulate molecular formation in a cloud of atomistic soup to see whether or not there was an algorithm or a heuristic that would guide accelerated bond formation, if that makes any sense. So that we found stochastic hill climbing to be optimal. And at that time, I showed Dave Deemer, who I'd met just 2 years before, a little gizmo that I had made to try to start simulating this with chemistry, because I realized simulating it with computers were orders of magnitude off for compute. And he said, I want to show you something. And we came down the hall, and there was this instrument that was an aluminum disc in a plexiglass chamber that would turn, and it would subject different chemical experiments to wet and drying. And it was like a huge version of what I had thought to do. And so that our partnership really kicked into high gear. And for the next 10 years, we developed the hot spring hypothesis for an origin of life, published it in a preliminary form in 2015, and then it was on the cover of Astrobiology Journal in 2020, and has been sort of a little bit instrumental, I would say, in the catalytic paradigm shift of the origin of life field from an ocean vent hypothesis, which really was more conjecture, because very little was able to be demonstrated chemically with a vent scenario from deep in the ocean, back to Darwin's warm little pond on land idea. Only in the 21st century, it's a hot little cycling puddle that has an engine called wet-dry synthesis, where you dry down and you re-wet. And in the last 3 or 4 years, I've been literally going to hot springs from New Zealand to Yellowstone to recently Fly Geyser at the Black Rock Desert in Nevada, crouching down and repeating our chemical laboratory experiments in hot spring environments and forming little things we call protocells. We're synthesizing or polymerizing RNA and DNA and getting them into little wrappers of lipid. Then they form these things called protocells, which can then undergo selection by their trillions.
I remember talking with you about this probably, I don't know, maybe 4 or so years ago. It's interesting because contemporarily computation led by some, pretty interesting research has moved considerably faster than I think we probably could have even speculated. The nature of ChatGPT and all the, the vast quantities of imagery and audio that computer simulation is now creating. Have you changed your view that maybe this might be something that could be computer simulatable?
Yes, to some degree, because during the height of COVID, we had a lot of visitors that were just coming to wanting to camp out on the land here at Ancient Oaks. And one of them was Ben Gertzel, who came with his family, and they just set up their camp here. And for several days, we got a chance to talk. And for those of you who don't know Ben, he is the father of AGI, the term itself, artificial general intelligence.
Certainly.
And he basically sort of elucidated how far they have got or not got. And basically, the AGI effort's really at step one. It's not even really underway. And that what we have in terms, he explained what we have in terms of AI now is these very narrow spectrum AI systems that aren't capable of general learning outside of the framework that they're trained on. So they certainly are not going to stand up virtually and start walking and making decisions. They're machines, they're machine tools. And AGI is much harder. So what Ben described, and you can actually listen to this brilliant talk he gave with Jim Rutt last year on the 3 paths to AGI. And the first path is cognitive, where you model the cognition of a brain from the abstract. And the second one is try to model the brain itself, neuron for neuron, neurotransmitter for neurotransmitter. And the third one is where we had a lot of concurrence, which is let's do chemical automata. And I know this is close to your heart as well, Tom, and some of our previous guests on Biota. And chemical automata would allow us to not have to model each molecular interaction as in neurons. It's just too challenging. But to model systems that are able to begin the process of Darwinian evolution and form networks and become general purpose learning systems. If you think of, microbes or humans as more general purpose learning systems, certainly not universal learning systems, he believes that sort of that middle ground is where we actually should start. And in the Genesis Engine project, which we've just launched with SETI Institute, literally as of a week and a half ago, the idea is to build simulation chambers where we can place these sort of colonies of polymers and protocells and have them migrate around, internetwork, compete, and witness, if we can in our lifetimes, the general purpose learning chemical automaton population grow up. And this is a little bit reminiscent of Noble Ape. And that system could be modeled in AGI alongside. And so Ben's nonprofit is actually launching a new project to do general purpose AI. And one of the case studies I think that he was expressed interest in is this chemical automata at the origin of life based on protocells and polymers. So that's a long-winded way of coming from the Netflix talk that we did, you and I, Tom, in 2011, all the way to 2021, where there's a final concurrence about how to do the digital side of modeling a chemical origin of life, meaning in the formality, the formalism of automata.
One of the things that strikes me, I've been playing with GPT-4, and as we are talking directly to long-standing listeners to this particular podcast, we have both Jeff Clune and Ken Stanley in the recordings. Ken Stanley now, I think, is listed as being the head of OpenAI's general AI. And Jeff Clune, I'm never sure where he is, but he's usually following in his footsteps. What's fascinating about what is seen now as a revolution is actually, as many of us know, it's taken many years in the process leading forward to these kind of things. The ability to parse vast quantities of data and then build upon that to make something which certainly the average YouTube video creator will say is intelligent and able to answer questions. And I was looking at GPT-4 with regards to poetry writing in the past couple of days. And it really is very interesting to compare what's going on with OpenAI, what Google is offering. I was playing with Ballard, which they just launched yesterday for most of the evening, particularly associated with the poetry and history-related problems that I like to pass these kind of things. I think the movement from artificial general intelligence to generative artificial intelligence, which is what these algorithms are now called, seems to be changing a lot of people's thinking with regards to computation, and really the neural models and these kind of things are secondary to the mapping of vast quantities of data and recalling that data very quickly. It's a different form of intelligence fundamentally, but it still certainly captivates humans. Have you thought about, I think what we offered with regards to the chemical automata discussion was certainly a very good building block for computation. But have you thought about utilizing the, vast quantities of memory and vast quantities of processing that contemporary computing has? Because I think that was the part of the Evo grid that always fascinated me, was the ability to get the right people together, the right kind of minds who were already developing this kind of code, and just make it vastly, rather than dealing with, small petri dishes, dealing with, potentially terabytes worth of data and potentially, teracycles worth of processing. Have you thought in that direction at all?
Yeah, and in fact, excuse me, what we're seeing really is that Darwinian selection of AI systems, of narrow AI systems, which generative as you're calling them, it happens when there are a lot of eyeballs wanting it. It has to appeal to people, but they swim in a sea of imagery and text and memes and things like that. That's their medium. So what we're trying to do with Origin of Life is swim in a medium of chemistry, which isn't that of much interest to your average person. So the actual ecosystem that artificial organisms are going to be living in is an ecosystem of human memes and human interests, really. So in some sense, our Origin of Life work is way off to the side because it's very specialized, because we need to simulate molecular dynamics so that we can go in and put the right chemicals into our starting mix and then sequence them. So we're totally tied to sort of biophysics metaphors, molecular dynamics metaphors. So in some ways, that's wet A life, but for dry A life or delicate silicon A life, whatnot, it really is this soup of memes. So one could actually really reconceive of the entire project now that we know that there's an ecosystem with foodstuffs and there are selective pressures around eyeballs, and we have plenty of users and plenty of drivers that you could instantiate an entirely new A-life movement within current generative AI systems.
Certainly. That's what I'd be looking towards. I think what was interesting when we talked about chemical automata those many years ago was the idea of simplified chemistry and using simplified chemistry and building on that. And actually, obviously, it leads itself from cellular automata. It's basically creating a similar set of rules, but mirroring what you're looking to do with chemistry. One of the things I found fascinating about the drying hypothesis, that basically what you had was cellular biology being generated out of these vast heating, cooling, drying, wetting environments. And that changed a lot of the potential with regards to cellular automata in my own thinking, because you almost became like a hard disk of information that was being constantly written to, read from, but in this chemical situation. How far have you gotten with DEMA associated with actually being able to simulate any element of that? You have computation on one side and you have then the chemistry on the other side. You're constantly returning to the chemistry as a means of moving the technology forward. What's your thinking with regards to that?
Well, we've had two collaborators that have done work on computation, and I've edited a number of papers submitted to our journal special issue where they're tackling the computational side. And the computational side tends to do the following. It tends to say, hey, you've got a population of, given that life has to start with polymers, really life in any form, but polymers they have to form naturally in some environment where they are formed just through, like in our case, the wet-dry synthesis. They don't have to have enzymes to stitch them together into long strings. And then what happens is a process of they start breaking down. And what's known as this kinetic trap is when the rate of synthesis of polymers exceeds the rate of their hydrolysis or their degradation. So you end up with more polymers and they end up elongating. And that's actually the fundamental core idea behind life itself. This is why you don't dissolve in the shower, Dave Deemer talks about, because there are enzymes constantly repairing all the breakages to your DNA and RNA and peptides, and you're constantly resynthesizing them because water is a universal solvent and will break it down. So different teams, one led by David Ross, the other Mason Hargrave out of our group, modeled the kinetic trap. So they were able to use MATLAB and other tools to really model the kinetic trap and then pair it with actual RNA polymerization and kinetic traps that we were actually seeing, we were actually observing. So that was very gratifying. Other groups actually model entire protocells in communication, or they model geochemical processes. And so that goes along, but it's extremely closely tied to the things you can test with experiment. So as far as how far along we've gotten, the great, in a sense, great kahuna of all this work is when these randomly synthesized wiggling polymers within envelopes of lipid, sometimes attached to the envelopes, are in such number and they're undergoing such number of interactions that you get a functional polymer popping out, something like what is described as a ribozyme, a piece of RNA that has a catalytic function that can actually do something, especially if it can make the building blocks for itself. So it would be an autocatalytic RNA ribozyme. And those are things that have been synthesized in the lab. That was the basis of the RNA world hypothesis, that RNA was sort of at the taproot. But it's also something that through molecular evolution, teams varying from Jerry Joyce at Scripps Institute to Bartel and Shostak at Harvard have shown columns of multi-hundred sequence RNA undergoing selection, and then spontaneous selection of an enzyme occurs, like a ribozyme capable of cleavage action is discovered and amplifies. So this has actually been demonstrated. So in our world of protocells, where we've got populations of different RNA, perhaps, and DNA and peptides, having function emerge, like a pore function, where there's a little hole in the membrane that allows things in and out, or where there's autocatalysis, where energy is absorbed and a product is made, or templating, where a piece of RNA templates a peptide or another piece of RNA. So you get a kind of quasi-gene. So all of those things are the next frontier in cycling chemical protocells through selection, is to see the emergence of clear functional polymers.
And in terms of SETI, historically, you and I have, I've seen you talk at SETI. Unfortunately, I missed the opportunity to talk there. How is SETI fitting into all of this?
So what's amazing is that, I've known Seth Shostak, the alien hunter over at SETI, for about 25 years. And I've been giving 7 SETI talks and SETI forums. And Dave and I decided to reach out to SETI over the summer and say, hey, we have a whole new thing going on with these Genesis engines, and it has a relationship to Frank Drake. And then Frank Drake passed away, I think, in August. And I was already giving talks about the Drake equation, the famous equation that gives you the proportion of worlds where intelligence might have arisen. And so I got in touch with them and say, look, we'd like to talk about Frank's legacy in terms of origin of life research, because the research is in a breakthrough phase, and it illuminates Frank's term F sub L, so the proportion of worlds where life can begin. And Dave and I and our colleague Francesca Carey, our graduate student, we had just published a paper called Urability, Factors on Worlds Where Life Can Start. So habitability is worlds where life as we know it might exist, might be able to survive. Urability is a world that can begin life, and it's really a different set of factors. And that's been proposed, and that was very attractive to SETI, because not only does it factor into the exoplanet research that they're doing, because it's, in a sense, you can now look at these exoplanets and say, well, they might be habitable, but can they begin life? And what Dave wrote to them was, for intelligent life to arise, it must first begin. So the F sub L part of Frank Drake's equation, we can actually put some numbers to it now. We can put some framework around it. And then what happened was we did the SETI talk. It was the first SETI talk in person in 3 years.
Mm.
And then 3 weeks or so later, Frank Drake's memorial was held, and we went down to the memorial. And there were a number of board of directors, people there. There was Frank's family were there. And it just spontaneously came up, hey, could we actually do a lab here at SETI? And one of the questions that Bill Diamond, who's SETI's CEO, had asked me during our SETI talk was, if you had $10 million, what would you build? That's a good thing for a CEO to ask of a nonprofit, who talks to trustees and donors all the time. And I said we would build a beautiful Genesis engine, an advanced engine that had literally a hot spring pool in it, had volcanic landscapes in the side of a chamber. And we would have injectors. We would have misters that would simulate fog and dew. We'd have injectors to simulate hydrothermal springs. And we would have wet-dry cycling. And then we would have a chemical analysis engine on the back end to look at what the protocells that were forming are doing. And then we would drive real-time gene sequencing using the nanopore sequencer that Dave came up with. And we would drive the whole system in a closed loop. And we'd have an AI engine on the side that would do predictions based on the polymers it was seeing, which ones were going to wiggle into functions, and it would tweak the system to try to steer it toward functional outcomes so that we didn't have to wait a million years or something. And that was very attractive to everyone. This was like fundamental science at the core of Frank Drake's equation, at the core of the question of, are we alone? And so literally, we had a meeting in December, and then we signed a memorandum of understanding about 3 weeks ago. And at a little ceremony, we had an interview with Shostak, Natalie Cabral, who's their Carl Sagan. She's really their chief scientist there, the Carl Sagan Institute. And just a wonderful thing. And we brought down our first version of that chamber, the chamber that I saw in Dave's lab back, in 2011 or 2010. And we installed it. We upgraded it. Our graduate student, Povilas Simonis, he's actually a postdoc. He's come from Lithuania. He's actually living here at Ancient Oaks. And he's now at SETI today running another battery of experiments. So the machine's been cranking for the last 2 weeks. And yesterday we met with their board of trustees to give a demo and a talk about the Genesis Lab project between Biota and SETI, so that they know it's something we're trying to raise funds to build the Engine 2 and Engine 3, those sorts of things.
One of the things I was looking up before actually reconvening to chat with you, Bruce, was your appearance on Joe Rogan's podcast, which was a favorite moment of mine in the time that we've spent together, because listening to you on Joe Rogan kind of at a high level was about finding the others, really, fundamentally. And I think that's been certainly, Terence McKenna's influence on you and these kind of things is incredibly important associated with finding the others. What do you foresee now associated with, what's the outreach means? Do you think you have the right team together? Do you want people to contact you? How do you find the others currently?
Currently, Tom, I'm just reaching out everywhere I can. I've been appearing on Jim Rutt's podcast. So 2 times last year, on Origin of Life. And Jim Rutt's an interesting guy. He was the board chair, I believe, but for the Santa Fe Institute. Mm. And so he has a huge Rolodex of people he knows. And he's an ebullient, great interviewer. And so we did Origin of Life and Origin of Life Implications, where we talked about Ben Goertzel and Whiteheadian philosophy and spiritual, even theological thinking, how that's impacted, all that sort of stuff. And then 3 weeks ago, or two weeks ago, another interview on a new subject that I'm working on, which is, can we use psychedelics? Now, this is relating back to Find the Others and Terence McKenna. Can certain genotypes, neurotypes use psychedelics to crack major problems in science, engineering, and leadership? And I've become obsessed with this topic because I've done that. I've used both endogenous, which is sort of internally generated, and exogenous, things added to your system to have creative breakthroughs. And one of the exogenous practices has been ayahuasca for me. And the entire Origin of Life scenario would not have been published in its fullest, most vivid, fully worked out nature based on my little brain working in a linear fashion. It had to come full throttle as a huge download over two mornings. Actually, one was in October 2013. The other one was the morning of December 30th, 2013. And that's when the entire thing just downloaded. The first event was ayahuasca fueled. The second was basically breath work, endogenous, and meditation. So I decided actually last year when Dennis McKenna invited me to a conference called ESPD in the UK, I decided to go public with my sort of psychedelics and high states of creativity practice, which I've now done for 20 years, and gave a talk about it. And then that talk has grown into a group of people that are wanting to support an international effort to bring back research in this area, which was last done by Willis Harman and Jim Fadiman and others in a publication in 1966 with the use of LSD and mescaline, actually, in creative problem solving. And this is talked about by Michael Pollan in his documentary series for Netflix. So literally, I decided to go sort of public with my own use and say, hey, can't we, all come out from under the covers, find the others who are microdosing or macrodosing in the corporate space, in the science academic space, in the leadership space. And this is a post-therapeutic application for psychedelics. But it's actually the original vision. So psychedelic was coined by Humphrey Osmond in 1957 to be mind manifesting. And into the early 60s, it was always thought that it was a tool for the intellect. Aldous Huxley talked about it in that way. And NASA was actually talking about using LSD to train the lunar astronauts for higher states of acuity and awareness. And this was in 1963. Then it hit the streets, it hit the youth culture, and all that kind of went away at that point. It went underground. So I've been, a week and a half ago, I was on Jim Rutt talking about this because it'll reach several 100,000 people. And that podcast will go live on Tuesday, this April 25th, 2023. And so there's a flood of interest that's coming back. I was at a conference for Bicycle Day this week up in San Francisco, and there were a line of people at the end of the talk about this who also are practitioners in private of these techniques.
Was the paper published in the Psychedelic Review in 1966?
Let's see. It wasn't in Psychedelic Review. I'd have to look it up. Which journal it was? It was a psych journal.
Yeah, I maintain a collection of those for, interesting occasional reading. It's part of actually my broader group of journals and books that you've inspired, particularly when you had Timothy Leary's library. Do you still have Timothy Leary's library, or did you donate it?
It has moved. It's not donated, but it's on loan to a psychedelic center in Portland, Oregon, where they're going to be scanning it.
Very good. Very good.
Yeah, they came. The DigiBarn also, it moved on long-term loan to Baltimore in 2020. Wow. 58,000 pounds of stuff moved to Baltimore, and it's now in a wonderful new museum project back there.
No, I remember going through, I think I put it on YouTube by exploration through the books, and from that ordered a series of books that I discovered in that library. I occasionally dabble back into it for inspiration associated with how, well, apparently at least free and creative things were prior to the illegality of it all. But it was fascinating to see just the breadth and dynamic academic literature that was, created, as you say, before things became considerably more illegal and locked down.
Well, in fact, if you look at the first article published in the first edition of Psychedelic Review in like May of '63, that first article, the first words are by Gerald Hurd, the philosopher Gerald Hurd. And that article was my big sort of Easter egg of inspiration. Well, the way he describes LSD and the way he describes its use in creative genius is the taproot. It's what I quote from when I do public talks on this. And hopefully this thing will become a movement. We're planning 5 articles over the summer for a big news outlet. And at the end of each article, we're going to offer a survey. So people can fill in the survey anonymously of their experience and their practice. And if we can develop what you would call a historical anecdotal database, then you can justify the research. You can justify that it's a thing. And that's how MDMA-assisted psychotherapy actually got justified for support. Government support was these surveys that showed people were using it for, say, anxiety disorders and things like this. So anyway, I'm hoping that all of this will become a thing, that I'm not necessarily leading. I'm more the Johnny Appleseed, but that it will grow and it will be able to emerge as legal psychedelic psychotherapy starts to roll out in the next 2 years. It'll emerge as something you can do alongside that or as a part of that.
Well, certainly what's changed since we last spoke is I've had twin daughters, and becoming a parent and experiencing that, but also experiencing the long process before actually, working through, and I've talked candidly with you about, what twins have been built based on. It's quite extraordinary that, these things can happen in any case. Now being a parent, I'm seeing early childhood wonder and exploration of spaces and exploration of self and a variety of other things come through that. And I think certainly as you are very well directed towards, psychedelics for use in research and development. And obviously, I mean, if you're familiar with the history of most of computing, the origins of psychedelic research are there, whether it be the history of Apple or a variety of other companies. But one of the things that fascinates me with my daughters is their ability to just pick up things and manipulate things and, make the world their own, so to speak. What is your plan for the next 5 years associated with your work? What will make the world your own in this sphere?
It's kind of a mishmash right now, Tom, and I'm hoping to do at least one more or several more Biota podcasts in the next few years. But in 5 years, I think we'll have annealed into something like this, that I'm working alongside reductionist materialist scientists. I'm also working with people who are in the realm of sort of high creative states, and that's very subjective and personal. But the third aspect is that the origin of life may be teaching us how biology actually rolls at the fundamental level. That biology, and this was conversations with a person you and I both know of their work pretty well, with Rupert Sheldrake. And Rupert Sheldrake's son, Merlin Sheldrake, was at the conference, Dennis McKenna's conference in the West Country last May. And he and I had a wonderful alcohol-fueled, typical British intellectual conversation that you might see at Cambridge and late at night. Mm-hmm. We had that at St. Giles' House, and his concept was the following: if you can grow these blobs, and that was his name for it, of protocells that are in chemical disequilibria, that are being showered with energy, that are moving and growing, they're sort of moving chemically into new possibility spaces or adjacent possibilities, as Stuart Kaufman would describe it. And if you have blobs that are next to each other, co-located with each other, is there some kind of non-local, bioelectric or proto-bioelectric effect that connects the two blobs that mean that they discover things faster, and versus a blob just on its own? And this is a test for his father's morphogenesis idea, morphoresonance idea.
Yes.
So Merlin had just published a book called Entangled Life that became an international bestseller, and he was kind of curious about what he was going to do next. So that was sort of a discussion opening that. And if you look at organizations like IONS, like the Institute of Noetic Sciences, what their scientists are doing is trying to verify that parapsychological phenomena are real, sort of remote viewing, sort of precognition of things about to happen, all those sorts of things, which are widely reported but very difficult to study and get statistically significant results in a controlled study. So that organization was founded by an astronaut named Edgar Mitchell 50 years ago. And somewhere in between, there's some kind of in-between space where the inquiry about high states of creativity, extraordinary states of sort of non-local connection of minds connects somehow to the blobs. That if they're non-locally connecting in a kind of field, a field effect that you can actually measure because the system's so simple, it's the starting of biology after all, then you can scale that up, learn about its properties and apply that to human experience. And that this may not be sort of solvable in my lifetime, but it's something that I think it's a natural place to take the blobs of the protocell work in its implications to the bigger picture. So that's, I think, in 5 years, if you find me a little bit less fuzzy about that, I would be most pleased.
Recruitment in these circumstances, I remember us having a few discussions, maybe candidly, maybe even recorded, associated with Richard Dawkins' problem of actually recruiting the next generation. You seem to be doing this with regards to undergraduate and graduate students. What's your feeling with regards to recruiting the next generation? I mean, obviously, Merlin Sheldrake is a good example of the next generation fundamentally. What is your thinking here?
What's happening in astrobiology, which is the field that I'm in, is an explosion. There are at least 30 new centers around the world, national centers that weren't there 5 to 10 years ago. And it's become sort of one of the hot areas of science, very interdisciplinary. You know, you've got geologists, computer scientists, even A-life researchers, chemists, geochemists, planetary scientists. It's a hugely interdisciplinary field. The conferences have been growing. The number of young people pouring into the field is pretty extraordinary. There's a lack of financial support for them, and that's what we're trying to do at Biota Institute is raise an endowment so that we can have a continuous mill of students and postdocs that come through. And with Povilas, his goal is to go back to Lithuania. He got a national grant to come here and work with us. And he'll go back and establish his own Origin of Life or Astrobiology Center. It would be the first one in Lithuania. And he'd raise his own funds and things. And then he would send us another generation of students to get training at our lab at UC Santa Cruz and at SETI. And then we would have 3 labs and 4 labs and 5 that are working on different aspects of the hot spring hypothesis, so that we're literally growing an entire generation of experimentalists and thinkers and computational people around a powerful new hypothesis, so that we have a better shot in our remaining lifetimes of getting it tested. The hypotheses are there to be tested, so that when, in 10, 20, 30 years, we can look back and say, we actually did a good job. This is a plausible scenario for how life can get started.
As folks who subscribe to this podcast are long-term fans of Biota in terms of the original vision, how do you describe the Biota Institute to that? Does it carry elements of the vision? What's carried over?
I think what's carried over primarily, the primary sort of core DNA of it was convening people of different walks of life, intellectual walks of life, backgrounds, as we did, as you know, at the Biota 1 conference, the Digital Burgess, and then it carried on from there in Cambridge with cyber biology, Digital Biota 2, and on to San Jose. And it was always like, can we get in the room a group of people who are fascinated about the question of life itself and what are emergent phenomena? Because emergent phenomena have got to be the conveners or the conveyors of magic, really. And if we can sort of crack that code, we can learn so much. And so we'd have all these different characters, people who are from the gaming world or people who were running deep sea drilling ships, or Richard Dawkins, who was at the second Biota conference. So that core DNA of convening different people around the question of life and emergence itself still carries forward. I think that where it's shifted in the last 10 years is a full turn toward wet A life, if you will, chemical automata models, rather than doing virtual environments where you're like Tierra, for example, Tom Bray's Tierra, where you're creating just an artificial environment to view virus formation, spontaneous virus formation and competition between gene strings, and seeing that that's actually possible in a simplified but just complex enough world. Moving sort of from that to focusing on a particular geophysical venue, which is hydrothermal fields, and having them teach us what is available, what the parameter set is, trying to get the molecules to do the walking, to convince our colleagues that this is actually plausible, and then reappearing in computing to ask, is computing of the 2020s with AI and with chemical automata, is it potent enough to help us along in our warm little pond simulations?
So when I put the message out to listeners, the list of people that they'd like me to get back on the Biota podcast was quite long. You were on there, obviously. We hope to talk with Jeffrey Ventrella and Steve Grant, speaking of digital biota too. So Steve has been, I think we've all kind of been somewhat hermit-like since we're involved with regards to biota. And Steve Grant in particular has been doing his own development, as was discussed in 2011. So it's going to be really interesting to have a chat with some of these folk and get a sense of where they are. And I think there's a kind of ongoing discussion associated with where the field is headed. And certainly Tim Taylor will also be appearing, my hope is. He continues to work with the International Society for Artificial Life. So I think there's a lot of potential in these podcast recordings to continue on the legacy of that in some regard. But Bruce, we'll definitely have to get you back on. Thank you very much for the opportunity to chat today. I know Saturday afternoons are precious things. They're certainly very precious for me as well. Any final thoughts, any final words for the podcast?
Yeah, I think that what would be absolutely wonderful, Biota really started I would say it really got going in August 97 with Digital Burgess, all of us meeting at the Banff Center and having the hike up to the Burgess Shale, which led to open minds and stiff and sore bodies. So then we went to a hot spring, which is kind of appropriate when I look back on that now. And then we sat in the Banff Center and we just kind of unraveled all of that group experience and all of our individual ideas about what emergence is, how life started on the Earth, how it complexified in places like as witnessed in the Burgess Shale. I think it'd be absolutely splendid if Biota could sponsor a meeting like that again in the next few years, where we have that diversity of people and where we go to an exciting place. I've recently been to Northwest Australia in the last 5 or 6 years to the ancient rock outcrops where they have found 3.5 billion year old evidence of both hot springs and microbes. And New Zealand is a fantastic place, also for sort of a view into all of this. But the thought I had was maybe Biota could sponsor a conference, for 50 to 100 people again, perhaps coming up on its 30th anniversary.
We're nearly there, right? We're pretty close.
We're pretty close. And it would just be lovely. And so if there's anyone out there who is in a position to help us do that, if you'd like Biota, is it 4 or 5? I think it would be Biota 5 or 4 to occur at some wonderful place around the world, and you want to help us make that happen. You have an institution or a way to do that. It would be wonderful to carry on that legacy, and for also people like as former, like the Jeff Clunes of the world and everyone that Tom is mentioning to come and give an update, especially from the interior, the cathedral of AI as it grows. And what is A life in the 21st century? We kind of know what it was in in the 20th. But where do we go forward? Sort of looking forward along all the directions of robotics, chemistry, origin of life, soft A life, AI A life, A life in space, A life for colonization. Does A life have a role inside AI?
I think it certainly does, and I'm really very pleased to have created such an archive of audio over however many years it's been, 20-odd years by now. Because if you look at what OpenAI is doing and, these kind of companies, to see who the founders of these companies or certainly the intellectual capacity of these companies was developing in terms of open source software a decade or so ago, it gives some pointers. And I'm not sure who the listener base is to this particular recording. I get the sense that maybe Herbert-like curmudgeons who have tinkered with computers at some stage might be a portion of it, as opposed to CEOs of large companies. But look, I will post this out to the listeners to challenge them to get back in contact and to indicate specifically where they'd like to see Biota 5 be held. Bruce, it's been far too long. It's been at least a couple of years since we've had a candid conversation. But I look forward to talking to you again in a similar format at some stage in the near future. Please do keep in contact. Please do let me know how things go. And look forward to talking to you sometime soon.
Thank you, Tom. The feeling's mutual.